Welcome back, everyone. Today we're going to talk about how pregnancy happens. So lecture 14 is fertilization, implantation, and embryogenesis. And in this clip, we're just going to go through 14A, which is fertilization. So when you think about it, you can represent life in three images, thanks to the oatmeal here for this cartoon. Penis in vagina. High fives. Babies out of vagina. Genes perpetuated. High fives, right? So the whole point of reproduction is to produce more of your species. So how do we make that happen? On the female side, certain preconditions have to be met, right? So we have to have a normal menstrual cycle. During the follicular phase, we have to be seeing development of ovarian follicles. Eventually, one of them is going to become fully mature. We're going to enter into a positive feedback loop with estrogen and LH, resulting in ovulation. That oocyte is going to be released, and then in order for this all to work, we have to also create a good corpus luteum that's going to create enough progesterone for the uterine lining to get nice and thick to receive that fertilized egg. I'd also like you to recall that during ovulation, the cervix tends to open and widen to make it easier for sperm, and the cervical mucus gets stringier and forms channels that help direct the sperm upward. Also recall that when the oocyte is ovulated and released, we have that thick protein coat called the zona pellucida surrounding the oocyte. We also have that grouping of cells, her besties, the corona radiata. So the oocyte is released, and then it has to be taken up by the fimbriae of the fallopian tube, right? With those little like finger-like projections where they have to kind of suck the oocyte into the fallopian tubes, where then the cilia will continue to waft it onward down toward the uterus. And there's all that information for you to review in case you didn't catch it. On the male side, we need to have sperm, right? So we need to have lots of sperm. We need to have usually somewhere between 250 and 500 million sperm per ejaculate is what's considered normal. They also have to be normally shaped. They have to have nice slim heads and good strong tails. Sometimes sperm are made that have two tails or just two heads or all kinds of strange shapes. So you need normal shape, which is normal morphology. They also have to be good swimmers. So sometimes sperm just kind of swim in circles. Sometimes they swim really slowly. We need to have good swimmers. The other thing is that they have to meet the egg before she dies. So the oocyte, once it's released, is only going to live for about somewhere in the order of 6 to 12 hours. Now, sperm can live much longer than that. Sperm can live somewhere on the range of 5 to 8 days. We know that they're most potent at potentially being able to fertilize an egg in the first 24 to 72 hours, but they can survive for up to 7 days. Not so for the oocyte. She can only live for about 6 to 12 hours. So the sperm either has to already be waiting in the fallopian tube for when the oocyte comes out, or it has to get there very quickly after ovulation occurs. And again, there's that information if you didn't catch all of it. Once the sperm reaches the egg, and so remember she has her besties, the corona radiata, surrounding the oocyte, they are going to burrow down in between the cells of the corona radiata, and then eventually the head of the sperm is going to make contact with the zona pellucida. When it does, it's going to release enzymes from its acrosomal cap, right? Remember that acrosome in the head of the sperm, so that it can actually digest through and dissolve away that zona pellucida so that it can get to the cell membrane of the oocyte on the inside. Once that happens, the egg will do a reaction that locks out any other potential sperm. As soon as one gets in, closed for business, no more sperm are allowed inside. The sperm that did get in, his head is going to open up and release the nucleus, which is going to combine with the nucleus of the oocyte, and together they are going to combine and form a brand new nucleus of a brand new cell. So now once the oocyte gets fertilized, we change the name of the cell. We now call it a zygote. Once those two nuclei, each with only 23 chromosomes, combine to form a normal diploid nucleus with 46 chromosomes, now we're going to call it a zygote. And again, there's that information for you. Once fertilization has occurred, the zygote, which is in the fallopian tube, because the oocyte hasn't had time to make it down to the ovary yet, right? So fertilization takes place in the fallopian tube. There's some additional events that are going to happen right away in that first week. First, after about 12 to 24 hours, that first cell, our zygote here, this is a zygote, is going to undergo mitosis. So remember, mitosis is a form of cell division where you create a new cell that has exactly the same number of chromosomes as the original cell. So the daughter cell is identical to the original cell. This is in contrast to meiosis, right, which was what we used to make germ cells or sex cells, where you ended up having the genetic material and increasing the diversity of the arrangement of genes. So cleavage, not that kind of cleavage, this kind of cleavage is a rapid mitosis that happens so that one cell is going to become two cells. And then those two cells are going to each reproduce, and then you're going to have 4 cells, and then you're going to have 8 cells, and then 16, and then 32, and you can see where I'm going. Eventually, you're going to end up with a ball of cells called a morula. Morula is the Latin word for raspberry, because somebody thought that little ball of cells looked like a raspberry. After you get to the morula stage, you're going to continue to divide and then form a hollow ball of cells called a blastocyst. This is going to happen around day 6 to 7, so about a week after sperm meets egg, and it's going to be the blastocyst that's going to implant in the uterus in the endometrial lining. The blastocyst is very interesting. Here is a summary of what we've talked about so far. We start off with our fertilized egg, the zygote. It's going to undergo cleavage and become a morula, or a ball of cells, and then it's going to turn into a blastocyst, which is a hollow ball of cells with a little clump of cells at one end. So the blastocyst, we now have, for the first time ever, what we call differentiation. So until this point, all of the cells, from the zygote to then when we had 4 cells and 8 cells, the morula, they all were exactly identical to each other. Not only in terms of their genes and their chromosomes, right, because every cell in your body has the same genes and chromosomes, but in terms of which genes are turned on or turned off. That changes when you form the blastocyst. This is the first fork in the road where cells realize, oh wait, I'm going to become this other thing and I'm not going to become one of these things. So we have 2 main components to the blastocyst. Here's our blastocyst here. This is a microscopic image of one here. First of all, we have the trophoblast. This is the outer shell of cells. The trophoblast cells, once they're in the trophoblast, now their fate is sealed. They're going to become part of the placenta and the other supporting organs of the pregnancy. They are not going to become the baby, which is kind of weird to think about, right? That your placenta has the same genes that you did, and yet after you were born it got thrown in the garbage. But that's the truth of the matter. So the trophoblast is going to become the placenta, and the inner cell mass, which is that clump of cells located at one side, you can see it here, the inner cell mass, that is what's going to become the embryo, which, if all goes well, will become the fetus and eventually the baby. So it's a pretty cute baby picture of you right there, a little clump of cells, right? So we all start as an inner cell mass. This is an image that just gives you an idea of that differentiation. So when you're a morula, you are what we call totipotent. Potent means power, toti means many or all, right? Like in total. So when you are still in the morula stage, any of those cells could become anything. They could become placenta or they could become part of the embryo and fetus, right? But then once you get to the blastocyst stage, now you've kind of picked your path. Either you're going to become part of the placenta or you're going to become part of the fetus. Those inner mass cells, that inner cell mass cells, are what we call pluripotent. Pluri is in plural as in many, because they can still become many different things, right? Some of those cells are going to go on to become heart cells, some are going to go on to be brain cells, some are going to go on to be immune system cells, some are going to be muscle cells, right? All the different cell types that you have in your body are going to form from cells that originally, ancestrally, were those inner cell mass. So if you've ever heard somebody talk about stem cells, what they're talking about is cells that are either totipotent or pluripotent, cells that haven't yet differentiated, cells that still have all the possibilities ahead of them. As you can imagine, that's really, really useful for researchers. Right? Imagine you have somebody whose brain cells aren't working well because of a disease. Well, hey, if you can get some stem cells and encourage them to turn into brain cells, perhaps then you can help treat that condition. So the reminder, up to this point, all of the cells have been totipotent, but now once we differentiate into trophoblast or inner cell mass, your fate is sealed. You're either going to become part of the placenta and its accessory structures or part of the embryo or in the future baby itself. So all of this is happening in the fallopian tube. So when ovulation happens, right, here's our oocyte, and she's not going to live very long. So the sperm have to get there pretty quickly in order to cause fertilization before the oocyte dies. The first couple of days, you're going to be undergoing cleavage while those cilia in the fallopian tubes go whoosh, 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 and waft the oocyte closer down to the uterus. By the time you get to about day 6, 7, right, you've already gone past the morula stage and you're entering the blastocyst stage, and then you're going to actually implant, and the blastocyst is going to move into that endometrial lining. So our summary. Fertilization, right? It's the process where sperm plus oocyte equal a zygote. 23 chromosomes plus 23 equals 46. The sperm we have to have good numbers of sperm. They have to be highly motile. They have to be good swimmers, normally shaped. And then they have to use the enzymes in their acrosomal cap to burrow through the zona pellucida. Fertilization is going to happen in the fallopian tube because the oocyte only lives for 6 to 12 hours. So if you really want to get pregnant, you want to have sex either a day before or within several hours of ovulation to give the sperm time to swim up there and meet the egg before it dies. Once fertilization occurs, that zygote is going to undergo cleavage, which is this process of rapid mitosis with doubling of the cell numbers, eventually forming a ball of cells called a morula, and then that ball of cells is going to hollow out in the middle. The outer part is going to be the trophoblast, which will become the placenta and some other structures, and then that inner clump of cells called the inner cell mass is what's going to become the embryo. All right, we'll pick up from there with 14B on implantation.